presented by the UT Knoxville Alumni Office. My name is Mallory Mendence, and I will be the moderator for the webinar today. Today, the webinar will focus on the basic biology of COVID-19. Next, let's go over a few ground rules for the webinar today. You notice that your mic has been muted. This will be the case for the duration of the webinar to avoid any disruption for our presenters. If you have a question or comment, please use the Q&A feature to share. Feel free to enter questions throughout the duration of the presentation. Questions or comments will be answered at the end of the presentation by Professor Becker. Please keep your contributions helpful and considerate to the host and other participants. Please note this webinar will be recorded and you are welcome to share it following the recording. Have fun. We look forward to bringing this topic to you today. We are so excited to have Professor Jeffery Becker with us today. Professor Becker is a Chancellor's Professor Emeritus and a former head of the Department of Microbiology at UT Knoxville. Professor Becker is a fellow of the American Academy of Microbiology and the American Association of the Advancement of Science. He has served on many NIH grant review panels, editorial boards of scientific journals, and as a consultant to many pharmaceutical companies. Welcome, Professor Becker. We thank you for being with us today.
Thank you, Mallory, for that lovely introduction, and I look very much forward to this. Okay, Vols, let's get started. We're talking about coronavirus and COVID-19. Here's a brief outline of my presentation. First, a little disclaimer. I've attempted to concentrate on a selection of issues dealing with the science of SARS-CoV-2, which is the coronavirus itself, and the disease it causes called COVID-19, named after coronavirus infectious disease 2019. There's so much information and controversy about this pandemic. I am trying to leave the political issues to the politicians. So I'll be explaining, and I'll use this cursor to show you where I am on the slide and subsequent slides. We're going to talk about the life cycle of the virus, which is really the basis for understanding everything about drug development and vaccines and its infectivity. We're talking about testing because it's such a controversial subject at this time. And I'll talk a little about the procedures and the reasoning behind the various tests. Then we'll go into the pathogenesis of the disease itself. And finally, most of the talk, or a great portion of the talk, will be talking about drugs, antiviral drugs, which we'll call therapy or treatment of the disease once it's started, and the coronavirus vaccines, which are to prevent. They're called prophylaxis from the disease. And finally, a short lesson, so to speak, on herd immunity. So I want to review the life cycle of the virus because really, again, this is really the basis for all our understanding. So a virus is an infectious agent. It's only able to multiply within the living cells of a host. So if a virus is on a surface or of any kind of surface or outside the host itself, it cannot multiply. It's really not living. It can only increase or replicate inside a living cell. Viruses have host range. That is, certain viruses infect certain individuals or species. So the coronavirus, the SARS-CoV-19, can only infect humans. There's a big controversy about where it came from. There's no doubt the scientists, scientifically, that it came from an animal transmission or a mutation of an animal virus that is closely related to the coronavirus that mutated and to now be able to infect the humans. And we'll talk about the basis for this host range in a few minutes. Viruses also have tissue specificity. So viruses can only attack certain tissues that have the mechanism or the receptors that recognize the virus. Now, this is a diagram of the coronavirus, the SARS-CoV-2. Notice that these yellow things, proteins, sticking out of the virus surface are the spike proteins. We'll be talking a lot about the spike proteins because the spike proteins are the basis for its attachment to cells, determining its tissue specificity and host range specificity. And the spike proteins are also really the basis for many of the vaccines that are being developed now. The virus genetic material is RNA, whereas most cells, our human cells, the genetic material is DNA, as we know. DNA carries all the information that the cells need to replicate itself. This particular virus has an RNA as its genetic material, which makes it different than most viruses and the host cells. And we'll talk about the significance of that. The virus also has a membrane that's actually derived from the host in which the virus replicated. So this membrane is the host membrane, the host cellular membrane, that does contain some host membrane proteins as well. And now the most important, the viral life cycle. So this is the host cell, a human cell. In our case, it's cells lining the lungs and other tissue that have on it receptors that bind are recognized by the virus or that the virus recognizes. So this shows the coronavirus attached to the host cell through its binding to the receptor. The receptor virus binding, that's a very important interaction that determines whether the virus can infect a cell. It determines the host range, it determines the tissue specificity as well. So that receptor is all important for the initial steps of the virus infectivity. After the virus binds to the receptor of a host cell, it enters the cell, and then once it enters the cell, it uncoats or it dissolves, and the RNA of the virus is released inside the host cell. The host cell doesn't see this viral RNA as a foreign RNA, it sees it as its own RNA. And what it does is then use that RNA of the virus to replicate itself and to make more proteins, and then finally to assemble into more virus. Now, this is really interesting because the virus is the ultimate parasite. It doesn't have its own machinery inside of it to do much. It uses all the machinery of the host. The cells, the virus are uncoated by host enzymes. The RNA is replicated by host enzymes. The protein synthesis machinery is the host protein synthesis machinery. And finally, it assembles sort of spontaneously to produce many, many viruses on release. So here's one of the things about viruses that is so insidious as well. One virus infecting a cell can release many, many progeny viruses. So you don't have to have many viruses infecting to get a very large proliferation. And then each one of these daughter viruses then can go infect another cell. And you can soon see how you can get a tremendous burst of viral particles by an initial infection. Notice that the virus is very small. It's 100 nanometers. That's a nanometer is 1 trillionth of a meter. So that's pretty small, cannot be seen by the naked eye. Which is one of the reasons why we really never understood viruses until they were first recognized by electron microscopy about 100 years ago or so. Now these small particles, this is a classical representation of a sneeze. This is not an unusual sneeze. This is sort of a normal sneeze that a person has. Can you imagine this sneeze releasing 200 million viruses into the atmosphere? And they're released in the forms of droplets, which can be released by coughing, sneezing, even talking and laughing, even loud talking. The louder you talk, apparently the more droplets are released. Singing, they can release as an aerosol as a smaller droplets that linger in the air longer than the large droplets. And of course, sputum and saliva contain the viral particles. And we're going to see how sputum and saliva is actually used to sample the virus in some of the tests. And these airborne transmission travel 3 feet, 5 feet, something like that. So this is one of the bases for social distancing, this sort of magic 6 feet. It's not really a magic 6 feet. It's an approximation of after 6 feet, then the amount of viruses in the air is relatively small. but they're so small and they're can be carried. So this is one of the reasons why we wear masks, to prevent this transmission through talking, laughing, sneezing, and so forth. So why do we need to test for virus? Well, there's many reasons why testing is very, very important. First of all, we would like to know who was infected, because this virus not only is infectious, that is, it can be transmitted to other people. When the infection is active in a person, this virus can also be transmitted when a person does not have any symptoms whatsoever. And it's thought that there's 4 or 5 days between a person contacting or contracting this virus and when it's actually symptomatic. So person can be perfectly healthy, feeling perfectly healthy, walking around, and yet still can be infected and can transmit the virus through these various mechanisms. So that's why it's good to quarantine a person that's actively infected or is asymptomatically infected, so that the virus will not be spread to others. And also, it would be great if we could do trace contacting, again, in order to test and possibly quarantine even those persons that were contacted an active or asymptomatic person. Certainly test those persons that were contacted to see if they are asymptomatic. The whole idea is to isolate the infected person or the asymptomatically or actively, so that it's not spread through the population. So we need to know the extent of infection of population. This affects many aspects of drug development, of vaccine development, understanding the spread, and so forth. So we might also like to know who has recovered from infection, because if a great amount of the population has recovered from infection, this contributes to the general overall ability of the infection to spread in the population. We'll talk about that later when we talk about herd immunity, which is a very important concept in this whole COVID-19 pandemic. So this is a little diagram that shows a person in red that's infected or asymptomatic. That person may spread it to several other people, or even potentially not spread it. We don't really fully understand the whole aspect of the contagion. We do know that the coronavirus is particularly more contagious than many other viruses that have been encountered to date. It's much more contagious, for example, than the common flu or influenza virus. So we're going to get back to this later when we talk about herd immunity. So there's various tests that we talked about why we want to test. Now I'm going to talk about the methods of testing and the procedures. The main test that's very accurate that can measure a current infection, whether it's symptomatic or asymptomatic, is to test for viral RNA. So RNA, again, is that genetic material of the virus. A swab can be taken, and there are nice tests that are applicable and very accurate. I'll show you that in a minute. The value of that test is to start treatment. Of course, right now we really don't have any treatment, but if we have treatment, it will be started then when it's detected, to isolate or quarantine that person that has an active infection, and then optimally to initiate contact tracing. There's 2 other tests that are much less effective, and in fact, antibody tests are widely spread now. Antigen tests less so. An antigen test tests for a portion of the virus in the body. So, for example, the spike protein is a portion of the coronavirus, as we saw on that diagram. It's that protein that sticks out of the surface that's recognized by the receptor on the host. So that antigen, if it's detected, it says that that person probably has an infection of the virus. So that measures current infection. It's very, very rapid. It can take place within a matter of hours. However, it's very poor in its both positive and negative readouts. Again, if that test is accurate, it can be used to start treatment. to isolate or quarantine, and to initiate contact tracing. And then finally, the antibody test. So the antibody measures the body's response. It doesn't measure the immediate infection because it takes approximately a couple of weeks for the body to make antibodies, for the humans to make antibodies against a viral infection. So the antibody test only measures past exposure or an active infection. But the infection could have been there for 2 weeks before you even detect antibody. But the problem with the current antibody tests is they're very poor. They give a very high false negative and false positive. So they're really almost no use at all, even though they're out there in the public. You can buy them from CVS or Walgreens, but they're very poor. One needs about a 99% accuracy, which is a very high rate of accuracy, for an antibody test to be any good. The antibody may indicate recovery because if you have antibodies and you don't have an active infection, it means your body recognizes virus and made antibodies. It also could be a source of convalescent serum. So there's some argument then, taking the serum of a person's recovered, isolating the antibodies from that person, and using that antibody to treat a person with an active infection. It's a very expensive treatment, but perhaps as a possibility in late stages and a person was highly infected with the COVID-19. And also, it's very important for evaluation of vaccines to measure the antibodies response. In the case of evaluation of vaccines, those antibody tests are very good because they're done with high precision. But the common ones in practice now are really not worthwhile. So this is the antibody test for RNA. This is the original test for the presence of virus RNA. It's called PCR, or polymerase chain reaction. What is done is a sample is taken of this famous swab up the nose or down the throat. That swab is then put into a test tube where the RNA of the virus is extracted by the medium in the test tube. The RNA shown in this diagram is amplified many hundreds of thousands, millions of times by this reaction called the polymerase chain reaction. And then those billions of copies of RNA are readily detected by a fluorescent dye. And if you get a bright indication, a fluorescent indication, it says that there was a viral particles in that swab collected in the patient. The instrument or machine that does PCR is a very simple instrument. This instrument is simply an instrument that raises the temperature and lowers the temperature, raises the temperature and lowers the temperature of the vials that are put in it to amplify the RNA. And this simple PCR instrument is probably in 50% of all biology labs in the world these days. It's a very commonly used instrument, and much larger versions are in hospitals everywhere. So it's sort of, really astounding to me and hard to understand why this PCR testing is not widely used, and it's very fast. You can get the results in a matter of hours. There's no reason to wait weeks. Only if if the swab or the medium has to be transferred to a large testing laboratory or something like that. PCR testing, though, is very accurate, very fast, and should be expanded rapidly because it can answer some of those questions posed above. The antibody or antigen test is much less accurate. It's first done by taking a small droplet of blood, putting that droplet in these little cartridges, which is about the size of this guy holding it in his hand right here. You put the drop in there, then the solution is added, and the antibodies travel up this cartridge. It's actually a filter paper by capillary action. And at the end, bands are indicated in that little test, whether they're positive or negative or have a control. But here's the problem. In a correct test for an antigen or antibody test, so this test we're looking at the antigen, we're looking at the spike protein. So we have on this little capillary instrument here, we have the capture antibody. So this antibody is manufactured, and it's put on this capillary and it captures the spike protein. And then you add to it a detection antibody that's fluorescent, or it's got a dye, and that is a correct test. So this shows that the spike protein is in the sample and it lights up where the detection antibody is. But you can get a false positive. In other words, there is no spike protein. But in this case, you have an interfering antibody that actually captured by that interacts with the capture antibody and the detection antibody to say, yes, there is the spike protein there, where it is not. And then you have a false negative where the spike protein is actually there, but you have an antibody that interferes with the interaction between the spike protein and the capture antibody. So here you have a false negative, here you have a false positive, and here you have the correct test. So the antibody and antigen tests are very poor. Now let's segue to the pathology of the coronavirus. So as the coronavirus is in the air, it's airborne, or it's on a surface and you get it on your hand, touch your mouth, nose, or possibly gets in through the eyes. It travels down the windpipe into the lungs and lodges in the lower portion of the lungs into the alveoli. So it enters the lungs are infected, the cells of the line can be infected and destroyed, both by the replication of the virus and by something else that happens a few steps later called the Cytochrome Storm. So the virus replicates in the lungs. Again, the lining of the lungs have cells that have the receptor for the virus on them. So the cells of the lining of the lungs are recognized by the virus. The virus enters those cells and replicates and makes more and more of itself. Well, the immune system doesn't like this. It sees it as a foreign object. So the immune system responds and makes antibodies. Not a lot at first, but it makes antibodies, and it takes a while to build up the antibodies. The antibodies then interact with the virus to try to destroy the virus. But if enough of the virus gets in there, the immune system is overwhelmed. And then the immune system does something that leads tremendously to the pathology of this disease. It actually attracts other molecules of the immune system, and it leads to what's called a cytokine storm. The cytokine storm then are all these immune factors. That then leads to further destruction of the cells of the lungs. The lungs cannot function properly. Fluid builds up the lungs, and it's literally the patient drowns because he cannot exchange oxygen. And in addition, not just the lungs, but we're seeing now that this virus can infect many, many other tissues of the body. So here is on the left is a cross section of the lungs, what it can do. Mucus builds up, fluid builds up in the lungs. It prevents exchange of oxygen. So the patient has to be put on a respirator to force oxygen into the lungs so you can breathe and get oxygen. Blood clots are formed in the heart and in blood vessel. This is a more and more a common factor of persons in late stages and later stages of the pathogenesis. In addition, it's been shown that the brain, the eyes, the nose, there's this very interesting pathological conditions where patients lose their sense of smell. The liver is attacked, kidneys, intestine. This is leads to a total failure of all your organs, and it's kind of like septic shock. It's very difficult to treat some persons with these later stages of coronavirus. Now let's talk about therapy. So a therapy is when you remediate or alleviate a health problem. It has to be, of course, diagnosed. And each therapy has its contraindications or its side effects. So there's many different types of therapies. We call drugs or therapies. But what really is optimal is to prevent the disease. So this is prophylaxis. That prevents the disease. And so what the role of vaccines are are to prevent the disease from happening in the first place. So with coronavirus, it's very interesting because there have been a number of a number of drugs that are used for viral diseases, although many of them are not that effective. But now what happened with coronaviruses is there's been a massive effort at what's called repurposing. So we take those drugs off the shelves of the pharmaceuticals and see whether or not they work for coronavirus. So here's some, again, back to this life cycle, which is the key to understanding the drug action. So this first step is very important, this reception between, or the receptor that recognizes a virus. If we can prevent that, we can prevent the virus right from the beginning. So maybe in very early stages of infection, if that can be diagnosed, the drug can be given to prevent these viruses from infecting other cells to magnify the disease. So there are some drugs that were developed for that. One is Fusion, which is the drug used for HIV, which is very effective. None of these drugs have been proven effective for the coronavirus because, again, this interaction between the spike protein and the receptor is very specific for coronavirus itself. Some drugs have been developed for uncoating, and here's a famous one, remdesivir, that was developed many years ago for viral replication, the RNA replication step. And it turns out that the company that developed remdesivir tested this early for coronavirus in a test tube and shown it was somewhat effective in the test tube. It's gone on to clinical trials and it's somewhat effective, but certainly not a miracle cure. But this is an old drug that was repurposed and is being repurposed probably more and more for the coronavirus infection. There are other drugs like that are for the protein production, Saquinir. There's this infamous hydrochloroquine. I won't get into that except to tell you that the recent studies have shown to be not only is it not effective, but it's dangerous. And finally, viral release. Tamiflu is a very good drug for flu. It prevents the release of the flu virus from the late steps and prevents its spread. But Tamiflu has been shown to be ineffective for coronavirus. So what's being developed now? So back to the attachment phase. So there's a drug, carnostat mesylate, that blocks attachment somewhat. It's not great, but it does a little bit. There's another drug called Lopinovir and a number of protease inhibitor drugs. The protease is important for the replication of the viral protein itself in its formation inside a host cell. And then remdesivir. These are drugs that are being developed now. And there are also other drugs that are very important that don't actually affect the viral life cycle itself. Now, we mentioned this cytokine storm, which is very important to the pathogenesis of the virus. So this drug here, which is a monoclonal antibody, actually tones down the release of cytokines, which is part of the immune system. And this drug has been thought of as a drug to use in later stages of the viral infection to affect the immune system itself, not directly the viral replication. And then there are antibodies against the spike protein that neutralize. Now, this is not a vaccine. These are preformed antibodies, even convalescent serum, that may be used to interact with the spike protein to prevent infection. So these are actually drugs, antibodies as drugs, not antibodies as vaccines necessarily. So let's talk about why it's been so hard to develop drugs for any virus, really. There are very few antiviral drugs. You know, when you go to the physician to get treated for a common cold, there's nothing much that can be done. Influenza, there's Tamiflu, which is only good in the first 5 days or so of the infection. There's very few other drugs on the market. Look at this. This is the normal pharmaceutical drug development cycle. You start out with thousands of drugs that work in the test tube. And then maybe some of them work in the test tube in culture against this virus you're looking for, or the bacterium or whatever. Out of that, only a few compounds pass that go into human trials. And there's 3 phases of human trials. A very small phase 1, where you only have 20 to 100 people in it, a larger phase 2, where you have larger numbers, and finally a phase 3, where you have thousands of people. And then it has to pass review, and you may get one drug out of 10,000 starting, maybe a million starting compound. And this is what the time scale is. Look at this. You start with a couple of years to get a lead compound. Those compounds are usually optimized by the medicinal chemist to get a compound that's better active or gets better lifetime in the blood. You have to do toxicology drugs, because a very important aspect of drugs is toxicology. And one of the important things about viral drugs is the toxic effect. Since the virus uses the host machinery, you have to have the drug has to be very carefully dialed so that it only inhibits the virus mainly and doesn't inhibit humans. You get all these side effects because the drugs are not totally specific. And then you have human phase 1, phase 2, phase 3 trials, and you're looking at 10 years normally to develop a drug for human medicine with costs approaching $1 billion. This is not easy, folks. This is really tough to develop a new drug. Vaccines are not much easier. So a vaccine is different. Again, this is prophylaxis. This is made from the whole virus or small amounts of the virus, and it prepares your body to fight the disease. We're talking about immunization, which is the process of getting vaccinated to make a vaccine. A vaccine are antibodies made by the human, made by yourself, immunoglobulins. They're Y-shaped proteins produced by cells of the immune system, and they're used by the immune system to neutralize pathogens such as bacteria and viruses. And the antibody recognizes a unique molecule of the pathogen called an antigen. So it's a interaction between an antibody and an antigen. Here's the Y-shaped antibody protein. An antigen are usually small molecules or parts of larger molecules that are recognized by an antigen binding site on the antibody. So there's an antigen-antibody interaction. In our case, the coronavirus, the spike protein or part of the spike protein will serve as an antigen to be recognized by an antibody. So antibodies attach to the virus. When they attach, they recruit cells of the immune system, white blood cells, macrophages that destroy the virus. However, if the viruses are rapidly growing and the active white cells are low, then the infection overwhelms the immune system, and this leads to obviously a very dangerous situation, pathology. So there's some vaccines that are interesting, and just for historical purposes and in comparison to what we're thinking of with the coronavirus. So since you're alums, I'm assuming many of you are in my age bracket, which is over 60. I won't say much more than that, but way over 60. And you probably remember the Saban and the Salk vaccines. So the Salk vaccine took the polio virus and it treated with a chemical to inactivate it, but it didn't change its protein structure. And those inactivated viruses were injected into persons giving a vaccine. Person developed immunity because antibodies developed against the outside of the virus and protected the person that was vaccinated with the Salk vaccine from a further attack from a real live virus. The Saban vaccine was similar, except it was an attenuated virus. In other words, it was a polio virus, was actually live, but its infectivity had been so dampened that it wouldn't cause an infection, but it would cause an antibody response. So these are the two classic vaccines. There are also many other vaccines that are listed here. There's a TB vaccine, there's a measles vaccine, and so forth and so on. Now with SARS, there are two major vaccine modes that are being developed. One is the spike protein. So you take the spike protein or a portion of the spike protein as the immunogen. That is what is injected into humans. And that spike protein then will elicit antibodies that hopefully will neutralize the virus. Another approach is this RNA virus, which is a very clever methodology. Here, this has never been developed for any virus or any infectious disease, an RNA virus. That's why it's so interesting and controversial. This is that you're reading about this practically every day. This is a company called Moderna, and Pfizer is also developing this RNA virus, where the RNA of the virus is injected into humans. That RNA is incorporated into the human cells, and the humans make the spike protein itself, and those spike proteins elicit antibodies against the whole virus. So this is only not the full viral RNA, because then you would cause an infection, but it's only that part of the RNA that encodes for the spike protein. So here is the antibody against the spike protein on the surface of the coronavirus. The antibody is attaching to the spike protein. Obviously, it neutralizes the spike or the virus from getting into the cell. Here's an electron micrograph of the coronavirus. The name came from its corona, its crown of spikes around itself. That's where the name came from. And here's a spike protein that has been crystallized and cloned. So we know the structure of the spike protein itself. So portions of the spike protein, not the whole thing because it's a big molecule, are either made by peptide synthesis, because this is a protein and portions of it are peptide, or part of the spike protein are cloned, the gene itself, to express in cell culture to make the protein a part of the protein. Now this is the DNA of the DNA virus, the DNA or RNA vaccines. There are many, many, many under development. Here, the RNA again is introduced into cells. The RNA or part of the RNA of the virus is expressed, the viral proteins are produced, and those viral proteins induce an antibody response, and the antibodies hopefully will protect against a subsequent viral invasion. This is how the antibody works. On the left side is a virus cell interaction. The virus attaches to the receptor, it gets into the cell, it releases its RNA, and more viruses are made. But if the virus is neutralized by the antibodies will block that virus from getting into cells. Sometimes it will form a large aggregate. Even sometimes it will get into the cell with the virus, and the antibody will block the virus from releasing its RNA and forming. This is how the vaccine will work. So vaccines are tested before they're recommended. It's very important, however, vaccines are sometimes can be very tricky. There are vaccines that actually enhance disease, and that has to be ruled out for a vaccine to be safe. Vaccines also, with drugs, they start with very small clinical trials to see whether the vaccine is safe, to see whether there's an antibody formed. Does the immune system work? What is the correct dosage? Then you scale up to larger and larger tests. They're licensed by the FDA. They're recommended by the CDC. They're monitored very closely for safety and efficacy, even if they're recommended for use. So this is really a mind-blowing slide. On the left here is what normally takes 15 years to get a vaccine into distribution for human usage. And you see what it takes, because these are done in succession. You have the preclinical manufacture of the vaccine itself. It goes into human trials, phase 1, phase 2, and these are successive trials. And then you have to, if it's worked, you got to build a factory to make millions and hundreds of millions of dosage, then manufacture, it has to be approved. In this emergency vaccine, what's going on now is the clinical trials and actually building of the factories are happening simultaneously. So there's billions of dollars that are being invested in building factories before it even gets through phase 3 trials. So you can see there could be a tremendous investment by pharmaceuticals and government in getting this vaccine very quickly into human usage. And even then, we're talking about 3 to 4 years. If it happens in a year and a half, it's a miracle, so to speak. And what the worst thing would be to do is release a vaccine that's not 100% safe. Because if you release a vaccine that's even 1% not safe, you're going to kill a lot of people because you're introducing the disease into the person by the vaccine itself. So this, finally, I want to talk about herd immunity. So herd immunity is the protection of a population or group, which is achieved by reducing the chain of transmission of an infectious agent because the population is resistant to the infection, either through immunization, through vaccination, or through prior infection. So here on the top here, you have this guy in red, he's infected. Here you have a population in which are not immunized or not immune, they're healthy. And now what happens when no one's immunized? What you get is this unbelievable spread from this one person to his or her neighbors, from her neighbors to other neighbors. And so now you get a huge amount of the population affected. If, however, you have some people in the population that are either healthy or in some way immunized or previously infected, the spread is lowered. So there's less red guys here. In a population where you have 60 or 70% that are immune in some way, the red guys here can not spread it to the blue guys because they're buffered by the yellow guys. And so here you have herd immunity, which we see in some diseases now, but certainly not in coronavirus. This is what we hope to do. And this also explains to some extent how testing and contact tracking or tracing is so important, because you want to be able to detect the persons that were contacted by the infected person. So those persons can be quarantined so they can't spread it to anybody else. And finally, just very quickly, I think we've run out of time. This is a comparison of the flu to COVID-19. COVID-19 is much more infectious. The hospitalization is much higher, and the fatality rate is much, much higher than flu. So it's a much more dangerous disease. So I think I've run out of time, and I would love to answer any questions that you have. So I see there's a nice question from Jenna. Thank you, Jenna, for your question. What is your take on why the virus seems to affect children less than adults? Well, you know, I don't have a take on that, because I don't think the experts have a take on it. It's very interesting, you know, whereas since we have so many childhood viral diseases, measles and mumps, and then which the children are the ones that are most infectious, there's been lots of speculation, but I don't think there's any really good answer to that question.
Thank you, Dr. Becker. I actually have an additional question.
Great.
So are there any summer activities that folks could, you know, partake in that would be safe at this point?
Oh, gosh. Well, I think everyone should wear a mask. It's not a matter of a political statement. It's a matter of really being kind to your neighbor, doing the right thing about being responsible. You may be walking around with the virus and you're asymptomatic and you don't know whether you're spreading, and you should keep your social distance. You should wash your hands and be careful. I think if you can do all those things, it's relatively safe to go out, but I don't know. My wife and I are staying in her house for the next few weeks at least. I don't think it's quite safe to go out, but if you wear your mask and do social distancing, wash your hands, maybe that's okay.
Thank you, Dr. Becker. And we have a few more questions in the Q&A
Okay,
there.
hold on, let's see. Oh, good. How likely is herd immunity without a vaccine, also with so much isolation? Well, yeah, it's a great comment, Mark. It's not likely at all. Because let's say there's 2 million Americans or 1.5 million. Let's say that 1.5 million Americans are infected now by testing. Let's say that's only really 10% because they're testing. But let's say there's 15 million Americans that have been infected. Well, we have a population of 330 million Americans. In order to get herd immunity, you're going to have 280 million Americans that are either previously infected or vaccinated. We're nowhere near herd immunity, not anywhere near it. And so Andrew asks, is there a systematic way to get to herd immunity? Vaccine is the best systematic way. There's no question. You don't want people infected. I mean, it's not a matter of, it's just not safe. Christy asked the question, are there any extra precautions you should take if you find out someone at your local grocery store or restaurant tests positive for COVID that you recently put? Well, that's a great question. I know if you've been in contact with that person and you've stood next to 2 feet in the grocery aisle getting your nice apples or whatever, I would suggest that you should be tested. And I think hopefully there are ways to get tested in most cities in Tennessee now. Maria asked, our city has asked everyone, even if they have no symptoms or don't think they've been exposed, to get tested to see if they have the coronavirus to get a better picture of how widespread the disease is in their area. This seemed like a reasonable. Well, if the city has the capability of testing everyone, and it doesn't cost you anything, I don't think this is. I don't think in a large city this is practical. Certainly Knoxville, where I am, doesn't have the ability to test 100, 200,000, 300,000 people. No way. So Alma asked, with a positive antibody test, how would you check for false positive? Well, guess what, Alma? In the current situation, if the antibody test is 90% accurate, this is a non-intuitive answer, but this is shown by statistical analysis, that with a 90% accurate antibody test, which is what most of them are now, not much higher than that, there's a 68% chance that you are false positive. In other words, if you test positive with a 90% accurate test, there's a 68% chance that that positive test was false. Now you can take that antibody test over and over and over again, but I think you can get the same answer. So I think the only way to do is to get the real thing is the PCR swab test.
Thank you, Dr. Becker.
Very good questions. Thank you. Thank you, Alum.
To our audience, thank you for attending. You will receive a short survey tomorrow regarding your experience. Lastly, we hope you will join us for our next Checkerboard chat on Wednesday, June 17th. The webinar will focus on careers and how to get to work now. Thank you again for attending today. Have a great day and go Vols.